Welcome back on our video series for evolution. In this video, part 4, we're going to look at population genetics. We're going to start with reminding ourselves a little bit about natural selection. If nature selects for differences in populations, let's say for faster and faster cheetahs, or for different colored moths, or for longer and longer necks, like we saw in our natural selection video, what accounts for these differences? What's the underlying cause? Well, the answer is genetics. So in this video, we're going to look at population genetics. If individuals have slight differences, and those variations are due to different allele combinations, then changes in the allele makeup of a population is the source of change. We can call this microevolution. So I'll ask you this question. What is it that evolves if it's not individuals? Pause the video and see if you can fill in this blank. What did you come up with? How about populations? Populations evolve, not individuals. So we study population genetics, how genetic principles can apply to an entire population. Now, with that said, we often don't study entire populations because it's just too hard. So we can sample populations, and as long as we sample well, we can assume that what's true for our sample is true for the overall population. We'll talk more about sampling in the class. We've done it before, but we'll do it again. But I want to differentiate between macroevolution real quick and microevolution. Up to now, we've kind of been discussing macroevolution, the change in look of a species over time, more of the phenotypic, what do we see as species change. But underlying that is microevolution, the change in the composition of the gene pool over time. Now, I've been using some terms already that we need to slow down and think about. So let's define a few of these terms, and we'll start with species. A species, how we define it, is that it's a group of interbreeding individuals who can produce fertile offspring. For example, donkeys can reproduce with horses. They make a mule. So the question is, are a donkey and a horse in the same species? Well, there's a two-part criteria here. They are an interbreeding group. They can breed, but it turns out that mules are infertile. So by definition, a horse and a donkey are not the same species. They don't meet the criteria, although obviously they're closely related. Now let's move on. How about population? Seems like an obvious definition, but there's actually a little more to it. A population is all the members of a given species, that's why we had to define species first, in a given area. So it's pretty specific. For example, you could say all the largemouth bass in a pond would represent a population of largemouth bass. All of the white-tailed deer in the national forest that you're in would be a population of white-tailed deer. All the human beings in the classroom is a population of human beings. And gene pool. This is what we're going to spend a lot of time talking about. The entire genetic content of a population. Every allele. Well, it's not realistic to really study the entire genetic content of population, but we can discuss the entire genetic content for a specific trait, for a specific type of allele. So let's look at this idea a little bit more. What is a gene pool? Why is it important? We're going to look at the change in the composition of the gene pool. So what do we know? We know that within populations there are differences, and we know that between populations there are differences. So the question is, what's the source of the difference? Where's the variation come from within and between populations? For sexually reproducing organisms, there are 5 places that can introduce variation into this process. Of course, gene mutations, they're the original source of variation. They create the new alleles, the new genes that never existed before. We've talked earlier in the year about crossing over and how that leads to new gene combinations by shuffling linkage groups. Look back at those notes if you need to. Independent assortment during meiosis, where we assort which of our chromosomes that we're going to pass along through meiosis, so we can look back at our meiosis notes there also. And then, of course, the fertilization event, the combining of DNA information from 2 different parents, creating a novel combination for the offspring. And chromosomal mutations, which we learned about in our genetics unit, or just our past genetics unit. And so 2 through 5 really just shuffle the deck, whereas variation 1 here, the gene mutations, is the source of new alleles. But do you understand what a gene pool is yet? I'm not sure we've gotten our head around it yet. I certainly don't think we've explained it well enough, so let's look at this again. We're not going to be able to study the entire genetic content of a population, but we can discuss it for one trait. So here's an example. Imagine we have rabbits, and we have brown rabbits, and we have white rabbits. And let's say that we know through observation that the brown allele is dominant to the white allele. So that to be a white rabbit you have to carry 2 white genes, but to be a brown rabbit you could carry 2 brown genes, but we know the heterozygote rabbit would also be brown because brown is dominant. Now, I actually want you to visualize a pool. Imagine that all the individuals in this population of rabbits came to the pool and dropped off their alleles, dropped off their genes. That represents the gene pool for this population. Well, of course, there'll be more than 3 rabbits, so we'll add some alleles in there. And so there's our gene pool. And now we can calculate allele frequencies. What portion of the gene pool is made of a certain allele? So looking at this gene pool, let's calculate the frequency of the dominant allele, the brown allele. Stop the video and see if you can figure out that calculation. Did you get it? There are 18 brown alleles and 30 total. So dividing that, we see that the frequency is 0.6 or 60%. How about for the recessive allele? Pause the video and figure that out. Well, hopefully you got that one a little faster. 12 out of 30 is 0.4 or 40%, or you could have realized that it has to add up to 100%. The total, the sum of the allele frequencies in the gene pool is equal to 1. So, if we let the frequency of the dominant allele equal P, and the frequency of the recessive allele equal Q, then P plus Q has to equal 1, or the whole gene pool. Well, how about genotype frequencies? What portion of the population is made up of a certain genotype? To make a genotype, I need 2 alleles. So if I were to reach in this pool and grab an allele, I might grab a brown allele. Or I might grab a white allele. The question is, do I have an equal chance of grabbing a brown versus a white allele? Well, in this case, I don't, because 60% of the alleles are brown and only 40% are white. If I reach in there a second time for my second allele to make a genotype, again, I have a better chance of getting a brown than a white. So if we were to set up a Punnett square to kind of represent this model, we know that there are 3 different possible genotypes that we can get. And if we were making a cross between 2 individuals here, we'd say that there's a 25% of getting homozygous dominant. But we're not talking about individuals, we're talking about the population. And for this gene pool, it doesn't work out that way because we have a much better chance of getting 2 big B's than we do 2 little b's, because there are more big B's in the gene pool. So in fact, we can represent this way, that the homozygous dominant genotype is P squared. It's a combined event. The probability that I pull the first brown and then the probability I'll pull a second brown is the product of the two events separately. So if I pull on the first pull a white and then the second pull a brown, that's going to happen 24% of the time, and I could pull a brown and then a white. Whoops, that'll happen 24% of the time, and only 16% of the time would I pull 2 white alleles. So we're going to represent the frequency of the homozygous dominant genotype with P squared. We can see that here. And the frequency of the homozygous recessive genotype is Q squared. So what does that make the frequency of the heterozygous genotype? Well, it's tempting to say P times Q, but there's 2 different ways to get P times Q. So that's 2 times PQ. So that if we add these all up, all of the homozygous dominants plus all the heterozygotes plus all the homozygous recessives has to equal, that's right, 1, the entire population. So we can represent this mathematically this way, our allele frequencies and our genotype frequencies. These are very helpful formulas that we're going to use a lot in class to solve problems. And this brings us to the Hardy-Weinberg principle, and I'm going to leave this over here because it's going to be helpful. The Hardy-Weinberg principle says that under certain conditions, the frequency of the dominant allele and the frequency of the recessive allele will remain unchanged from generation to generation. In other words, the gene pool won't change. We're at genetic equilibrium. No microevolution, therefore no macroevolution, therefore no evolution. Well, This is interesting, and it should be begging a question, and the question is, under what conditions? The Hardy-Weinberg principle has 5 conditions, and they are: no mutations can occur, the population must be large, mating must be random, in other words, no sexual selection, no individual may enter or leave the population, immigration and emigration, and no one genotype is more suited for survival than any other genotype, no natural selection. Well, the question is, does this ever happen? Even in a laboratory setting, could you control for all these variables? Well, the answer is no, of course not. So what's good is this principle? Well, it gives us kind of a null hypothesis. If we do see change, it's because one of these 5 conditions, one or more of these 5 conditions, has been violated. So how does violating each of these conditions lead to changes in the gene pool? You'll have to come back for part 2 to answer that. I don't want this to run over too much over 15 minutes, and so I'm going to break it in half right here. I don't think it'll be 20 minutes total, but this is a good place to break. But you must come back for part 2. I'll put a link down here. Part 2 is where we're going to get into the details of how to do Hardy-Weinberg problems, and again, to see how those 5 conditions lead to change. So please come back for part 2 on population genetics so you are prepared for class. And I hope you learned something. I'll be right back.